Hello, everyone. Welcome to the Emerging Trends in Pharmaceutical and Biopharmaceutical Analysis. I'm Jerome Workman, the Executive Editor of Spectroscopy, and I'll be your moderator for this event. We are pleased to bring you this afternoon session, which is part of a two-day virtual symposium presented by Spectroscopy, the Society for Applied Spectroscopy, and the Koblenz Society. The Society for Applied Spectroscopy, or SAS, is a nonprofit organization dedicated to the dissemination of information related to spectroscopy. SAS is pleased to be the publisher of the internationally recognized peer-reviewed journal Applied Spectroscopy. The Koblenz Society is a nonprofit organization whose purpose is to foster the understanding and application of vibrational spectroscopy. Both societies have an educational mission, and this event is part of that mission. We invite you to visit both societies' websites to learn more. This virtual symposium is sponsored by CEM, PerkinElmer, Thermo Fisher, and Metrome, and Endress and Hauser. And now a statement from our sponsors. Today's event would not be possible without the support of our sponsors. CEM Corporation began manufacturing analytical instruments in 1978. In 1985, CEM developed the first microwave digestion system, revolutionizing sample preparation for trace metals analysis. Since then, continuous innovation has kept CEM at the forefront of sample preparation. With worldwide headquarters located just outside of Charlotte, North Carolina, CEM operates 7 subsidiaries as well as a large dealer network covering the globe. CEM truly is the global leader in microwave sample preparation. At Metrome USA, we're more than a leader in chemical analysis. We're a community driven by passion, integrity, and a commitment to quality and innovation. With expertise in both laboratory and process systems for titration, ion chromatography, electrochemistry, and spectroscopy, our approach goes far beyond just technology. Discover how our dedication to both science and humanity shapes everything we do at www.metrome.com. Customers worldwide trust Thermo Fisher tools and solutions to help them accelerate innovation and enhance productivity. Together, they are making advancements that truly matter. Thermo Fisher achieves this by providing an unmatched combination of innovative technologies, purchasing convenience, and comprehensive support. PerkinElmer is a global provider of scientific solutions with an 80-plus year history of bringing expertise, innovation, and technology to our customers. They manufacture the latest in analytical instrumentation and provide essential lab support services to streamline workflows. Partnering with scientists, clinicians, and lab technicians, they accelerate scientific outcomes and aid the development of groundbreaking products. PerkinElmer believes in the power of science towards a healthier, safer, and more sustainable world. Endress and Hauser provides advanced measurement and automation solutions for industrial processes across the globe. Their offerings include instruments for flow, level, pressure, temperature, and liquid and gas analysis. With a focus on innovation, reliability, and digital integration, they serve a plethora of industries. Their portfolio comprises of many products for any application, from basic to highly specialized, ensuring efficient and sustainable process control. We have a few important announcements before we begin. This symposium is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found at the bottom of your media player. Please note that all questions will be answered offline after today's session. So when you type in your question, please include the name of the speaker that your question is for. I would now like to introduce our speaker, Patrick Brown. Patrick Brown graduated with a master's in biomedical science from the Pennsylvania State University and an MBA from the University of Delaware. He now leads the UV Viz product management team for Thermo Fisher Scientific. In his role, he works with his team to collect user feedback, integrate feedback into software and hardware updates, and works with marketing teams to communicate these benefits to the scientists that trust Thermo Fisher Scientific instruments. Patrick, thank you for joining us today. Please get us started.
Thank you for joining me today. My name is Patrick Brown. I lead the UVViz product management team within Thermo Fisher Scientific. And today I wanted to talk to you about our Nanodrop Ultra microvolume UVViz spectrophotometers and fluorometers. There are 4 Nanodrop Ultra instruments in our portfolio. The Nanodrop Ultra is just an instrument for measuring absorbance on the pedestal. The Nanodrop Ultra C provides you the option of the microvolume pedestal technology, as well as a cuvette port for measuring absorbance. Nanodrop Ultra FL is an instrument that measures both absorbance and fluorescence on the pedestal technology. And then finally, the Nanodrop Ultra CFL measures absorbance on the pedestal and through the cuvette port, as well as measuring fluorescence on the pedestal. You can see the pedestal technology in the lower left-hand corner, along with the LEDs that we use for the fluorescence excitation. One other feature noted here is an LED bar underneath the high definition touchscreen that indicates different instrument statuses, and that will change colors depending on the status of the instrument. So there are several key updates on the Nanodrop Ultra. Certainly one of the most notable is the addition of blue and red LEDs to allow users to measure fluorescence in addition to absorbance on the pedestal. There is an improved lower detection limit. The instrument can now measure down to 1 nanogram per microliter of double-stranded DNA. We know many users of the Nanodrop instrument do so within a qPCR workflow. So we have added an onboard qPCR recipe calculator to aid users in creating that qPCR reaction. We have updated our Thermo Scientific Aclaro sample intelligence technology such that the contaminant identification software can now distinguish between DNA and RNA for plant and bacteria sources of the nucleic acid. We have Thermo Scientific Cyvault 2 software, which users can use to enable 21 CFR Part 11 compliance, and that can be run directly on the instrument itself or attached to an optional companion PC. We also have Aclaro Pro software, which enhances the accuracy of the Nanodrop Ultra measurements across the entire absorbance range, such that measurements can be had within 5% error. And we have improved data export, including export directly to the Microsoft OneDrive, Google Drive, and Thermo Fisher Connect cloud platforms. Additionally, for connecting to LIMS, we have a RESTful API. Talking a bit about the specifications, you can see a lot of what I had mentioned here. One additional thing to note is that there is an optional the opportunity to use an external battery pack to power the Nanodrop Ultra. And that's because on the back of the instrument is a USB-C port that is used for power. So that would be found here. And so because of that, the instrument can be run off of a external battery pack that would be typically used to run a laptop. Turning the instrument on can be achieved by clicking the on button on the right-hand side of the instrument. And then up front, we have a USB-C port for data exchange, getting data off of the instrument. And for those labs that have not converted to computers or laptops with a USB-C port, we do offer with every single NanoDrop Ultra a dual USB-C, USB-A drive so that you can plug the USB-C side into the NanoDrop Ultra and then take that data back to your computer that might still be running USB-A. Another notable piece on the back of the instrument is this Wi-Fi dongle. So every instrument also ships with this USB Wi-Fi dongle. And so you can choose whether to plug that into the back of the instrument. Plugging it in allows you to connect the instrument to Wi-Fi, as well as Bluetooth devices such as a Bluetooth mouse or a Bluetooth keyboard. Getting to the fluorescence part of the instrument is able to detect DNA and RNA using our NanoDrop Ultra DNA BR fluorescence assay or the NanoDrop Ultra RNA fluorescence assay. And you can see the ranges of detection on the bottom right-hand side. So RNA and DNA can both be detected with their respective fluorescence assay from an initial sample concentration range of 10 to 1,000 nanograms per microliter. We also have what you see here, a fluorometer application. And this application allows you to measure alternative dyes that are outside of our kind of pre-programmed methods. And so here are the specifications for excitation and emission. The blue excitation range is 430 to 495 nanometers. Blue emission range would be 520 to 600 nanometers. And then red excitation would be 600 to 645 nanometers. And the red emission range is from 660 to 680 nanometers. Now, this reagent calculator, it is specific for our DNA and RNA assays. And this reagent calculator asks you to input things like the number of standards that you're going to use, the number of samples you wish to measure, and then aids you in preparing those assay reactions. The other notable kit is our FL1 fluorescence verification kit. And this is used as a diagnostic tool just to confirm the performance and linearity of the red and blue LED. We will be coming out with a application note, and that is going to utilize the commercially available Invitrogen Quantit Picogreen dye. And using this dye in our application note, you can get accuracy and reproducible data for original sample concentrations under 1 nanogram per microliter of double-stranded DNA, making it more sensitive than our DNA absorbance measurements. Talking about the lower end of our absorbance range, we do have a lower detection limit that is, again, lower than our NanoDrop 1 instrument. So now the software will measure down to 0.02 absorbance units, and that is indicative of a DNA concentration of 1 nanogram per microliter. Now, there's no new measurement path lengths to establish this, but rather we have modified our absorbance algorithm at the low end to enhance our ability to detect signal from noise. So some of those applications are actually unchanged in their lower detection limits compared to the NanoDrop 1 instrument. And you can see the table of unaffected instruments in the bottom left, or rather unaffected applications in the bottom left. Talking a little bit about the qPCR recipe calculator. When customers are measuring data in the double-stranded DNA application, they can select from a pre-populated list of qPCR assays, or if they wish, users can also use our kit editor and enter the specific kit information for the kit that you're using for qPCR, and then anything that they save shows up in the drop-down menu. So what this will do, once you select the kit and make your measurements, the software will calculate the recommended steps to prepare your sample for the qPCR reaction. So the software essentially knows what concentration that particular kit requires for the reaction. It knows what volume of the nucleic acid the kit requires, the reaction requires, and it also will do any dilutions necessary for higher concentration samples. And so the final template concentration is based on the manufacturer's recommendation. But if you wish, you can edit it yourself. And so what we're seeing here in the center is that for sample 1, the software is recommending that the user take 20 microliters of their nucleic acid sample, add in 80 microliters of diluent to dilute their sample, and then take 1.2 microliters of that diluted sample, add it to about 3 microliters of water, and you'll get a final concentration of 0.5 nanograms per microliter in the final reaction. And we have tooltip available by clicking on this blue eye that will walk you through exactly what each column is meant to teach you within this software feature. So one of the advantages of the Eclairo sample contaminant identification technology is the ability to differentiate DNA from RNA. And so typically we just look at the 260 nanometer value when we're thinking about calculating a nucleic acid concentration. We might look at the 260-230 ratio or the 260-280 ratio to consider the contaminants that might be present, whether they're organic contaminants or protein contaminants. But these values don't really tell us anything about whether it's a DNA or an RNA that we're looking at. What we're able to do is use chemometrics to essentially identify the different spectral features between DNA and RNA when it's of the same source. So whether it's mammalian DNA, mammalian RNA, and now plant or bacteria. And we're able to look at those different spectral features in the range of about 270 to about 290. And you can see for pure DNA and pure RNA, there are just slight differences here. And chemometrics can help us establish or differentiate between the two. So here we actually have a screenshot of what that would look like. In this case, the sample is a rat DNA, and you can see the original spectrum in green, but this sample has a contamination of RNA such that the corrected concentration of about 61 nanograms per microliter is roughly 40% lower than the original concentration that was estimated to be about 100 nanograms per microliter. That's due to that contamination of RNA. Now, the same is true when you're measuring RNA. If you have a sample that's been contaminated by genomic DNA through the extraction process, that will be flagged as well. One of the exciting advances in the NanoDrop Ultra is SciVault 2 software, which allows users to connect their NanoDrop Ultra to their domain network and actually log into their instrument using your company's or your institute's username and password. So just like you would log into your computer or just like you would log into your email, you can log into your NanoDrop Ultra instrument directly on the user interface on the screen when you have SciVault 2 installed. There are two scenarios. So like I just described, you can kind of set it up to your network and use your email password. But if you wish not to have the instrument on your network, you wish it to be disconnected, then you can create unique usernames, unique passwords, and kind of manage them separately from your domain linked username and password. Also, you can connect your instrument to PC with a USB cable, and all your user management would then be done on the PC side using your computer's domain credentials. Thinking about the new diagnostics, so we still have the performance verification application, intensity check, and pedestal image check applications to verify the accuracy of the pedestal path lengths, ensure the spectrophotometer and the flash lamp are performing well, and the mechanism that we use to confirm the liquid column is established between the top and bottom pedestal is working well. We have new applications to test the performance of the cuvette port, as well as the LEDs, which I previously mentioned. And then we have a new algorithm as part of our Aclaro Pro software, and we have a diagnostic to verify that algorithm's performance as well. So NanoDrop instruments for the past many years have been beloved by scientists in the research space. And this new Aclaro Pro software really aids those researchers, or I should say those scientists, in a more manufacturing QA/QC type setting. Now, the Aclaro Pro software is an add-on available to NanoDrop Ultra instruments, and it unlocks high accuracy versions of our most popular absorbance applications. And so you can see that you can measure up to either 400 mgs per mil of IgG or up to about 18,000 nanograms per microliter of single-stranded DNA, oligonucleotides, all within about 5% error. So it does look, the software looks identical to the standard applications, and the workflow is the same. You still pipette 1 or 2 microliters onto the lower pedestal, lower the arm, and press measure. We have a more robust calibration in these applications, a more robust algorithm in these applications, and it allows for a much better performance for those scientists who need absolute performance, specifically again in the manufacturing line when your batch quality really matters. Thinking about that production space as well, we have a RESTful API system where you can have data that is automatically transferred to your LIMS software, whether that's a proprietary LIMS software or one of the off-the-shelf software available. And we have technical support that's able to assist you with documentation on setting up that connection. One of the other export features that we have established here is a preview functionality. And so what that allows you to do is you can check boxes next to the different parameters that you might want to include on your report or in your data export. So if you want the application name or the serial number, whether you want just the report data as shown in the table, or if you want spectra data like is shown on the right-hand side with these graphs, you can choose what is exported, and your preview will provide you a glimpse at what that output will look like. You can export data as a TSV, a CSV file, the Nanodrop Ultra experiment file, which allows you to import that into the other instruments, or you can export your data, your report rather, to a PDF format directly from the instrument. When you have an instrument connected to the internet, you're also going to get real-time alerts when a new software version is available. It's something that it shows up as a orange banner as well as an orange alert. And so what you're able to do then is download the update and go through that installation process. If, however, you prefer to keep your instrument on, running the legacy software and not update, you can simply choose to ignore the update and choose to hide the future update notifications. Wrapping up, there are a few accessories available for the Nanodrop Ultra. So this Wi-Fi stick that I described comes with every instrument. So does the LCD display cloth. You may choose to purchase a instrument dust cover or one of our IQ/OQ kits to ensure your instrument is performing within the operational specifications prior to use. I thank you for your time. And if there are any questions, I'll be happy to answer them. Thank you.
Thank you, Patrick, for that great talk. I would now like to introduce our final speaker of the day, Adam J. Hopkins. Dr. Adam J. Hopkins is the spectroscopy product manager for handheld and laboratory instruments at Metrome USA since 2016. He has a PhD in chemistry from the University of Oregon, where he studied nonlinear optical spectroscopy under Geraldine Richmond. Between graduate school and Metrome, Adam worked developing spectroscopic solutions for standoff and non-contact sensing applications using LIBS, Raman, and other novel techniques. Dr. Hopkins is a member of the American Chemical Society, the Society for Applied Spectroscopy, the Covalent Society, and SPIE. He is the author of over 20 peer-reviewed papers and proceedings and 3 patents. Adam, thank you for joining us today. You have the floor.
Good afternoon. Thank you for joining me. My name is Adam Hopkins, and I'm the spectroscopy product manager at Metrome USA. And I'm going to talk to you a little bit about a new product line of ours, the iRaman NXG and the Duo, and the benefits that these systems can bring to you in the biopharmaceutical and biomedical research and development world. So if you're here, you probably know about using Raman for biomedical and biopharmaceutical applications. Probably the most common application for Raman spectroscopy in the pharmaceutical world is the identification of materials. Think dyes like OPA that might be used for solid dosage forms, various excipients, so polysorbates 20 and 80 in the liquid side, or various cellulose forms, even different cell culture media and lines, they can be identified with Raman spectroscopy. And the iRaman NXG adds the ability to make identifications through opaque materials in a pretty small form factor. You can see your probe is fiber optically coupled into the device. So these are your most common applications in the world of pharmaceuticals and biopharmaceuticals. But what else, right, is out there, right, beyond sort of this raw material identification piece? Well, we can monitor fermentation and reaction processes. So here is a simulation, real data, but a simulation of the consumption of glucose and the production of lactate in a fermentation. And at the top spectrum, labeled figure A, you can see the raw data that we're measuring. And then we process the data and build PLS models off of that, for example, to track what happens as we add glucose, and maybe then that is consumed, producing lactate, and then we feed again and hit some kind of steady state. So we can use Raman for monitoring fermentation and reaction processes in the pharmaceutical space. But there's more than that. If we think about biomedical diagnostics, right, and the power of Raman, we can meet demands for near real-time and minimally invasive analysis because we can be sensitive to molecular changes that are associated with disease. And the small footprint of the iRaman line supports sort of point of care analyses and diagnostic developments. Some examples that we might have would be, for example, biopsies for analysis of tissue samples, drug efficacy studies, identification of biomarkers in the early stage disease detection, even surgical targets. And we can even talk about deploying SIRS to provide signal enhancements and aid in trace level detection of disease biomarkers. Ultimately, right, there's a lot of different functional groups related to biomedical testing that have characteristic Raman frequencies. And we can see, all of those different groups in Raman data. So we can look at CH stretches, right, in the sort of the 4,000 to 2,500 wave number region. These are seen, but you've got your triple bonds, you've got your double bonds like CCs, and even complex patterns that are occurring in the fingerprint regions like I've seen here. And you can start to decompose all of this to get much higher level of insight into whatever system that you're studying. So I mentioned, diagnostics, right? And so here's an example, and I'm using this example for cancer margin analysis, and right from some collaborations. And right, pathology analysis, if you've ever been through cancer diagnosis, can take 1 to 2 weeks, oftentimes to do. But in this case, these researchers were able to use an older generation iRaman system to build classification models to actually identify normal tissue and cancerous tissue. And you could then use that to predict new tissues and then actually identify if you say removed all of the cancerous tissue from the edge of whatever you're removing. And that's what we're showing here, right? We've got cancerous tissue areas identified in red, built on the classifier, and then we have non-cancerous tissues, right, that are classified green, and then you have mixed areas where you're in the boundary. So we're able to build these kinds of diagnostics and look at them. But one of the things that is challenging when we're playing in the world of any kind of biological material is getting all of the information. Here we're looking, I've got 785 nanometer Raman spectra up here. These are covering about 400 to 3,200 wave numbers. If we want to maybe have lower fluorescence, we might move to a different wavelength, say 1064 nanometers. But now we've gotten rid of any of the CH, NH, OH stretching information in exchange for getting, higher signals, if you will, right, reduced fluorescence background in the fingerprint region. So we can maybe get a little bit more data in one end, but we gave it up on the other side when we move downstream. And we set about trying to say, well, how can we avoid that, right? How can we get everything that you want, but still keep the same small footprint for point of care diagnostics or other uses for a Raman spectrometer, because there's lots of cases where you want the CH, NH, and OH stretching region information. And so to solve that problem, we have developed a new line of Raman spectrometers, the iRaman NXG and Duo systems. And these have really been designed for today's research and development labs. They make precise measurements in seconds with high throughput Raman spectroscopy. Our new software allows you to really develop methods and create very streamlined quantitative and qualitative workflows. We'll show you a wide range of accessories, and these are available in standard 532, 785, and 1064 nanometer lasers, as well as the new Duo that extends the range for more comprehensive sample information. So the iRaman Duo allows you to access both the fingerprint and the stretch region. So we use two lasers in the iRaman Duo. We use a 1064 laser to access the, call it 200 to 2600 wave number fingerprint region. That's going to get you that detailed functional group and structural information that you're looking for in whatever your analytical system is. And then we added in, just added in another laser, an 860 nanometer laser, to give us the stretching region from 2600 to 4600 wave numbers. And that's going to give us access to CH stretches, OH stretches, NH stretches, and even hydrogen itself, HH. And this lets us get insight into water and how it's bonding in these biomedical and biopharmaceutical systems, as well as provide concurring data or collaborative data that matches what you're seeing in the fingerprint region, or provides additional explanation of what you're getting in the fingerprint region with the spectrum. And this is all in one box, no moving parts. We're doing it all with optical filtering on a single detector. The total wave number range of this system is about 100 to 4600 wave numbers, better than 10 wave number resolution. And again, I can't emphasize this enough. There's no moving parts, there's no complex optical setup, and you have just a single simple fiber optically coupled probe that you use for your data collection. The NXG is available with a wide range of our sampling accessories, the NXG and Duo. So vial analysis, cuvette, if you want higher analysis, direct immersion, see-through, and probe holders and microscopes, of course not shown on here, to allow for really intense investigations in whatever kind of sample setup you want to understand. We've got our new Spec Suite software. This is our new all-in-one software. So if you are familiar with this in the past, you know that you use different pieces of software to do different types of analysis. We have reimagined that, taking feedback to create all-in-one software that offers both exploration tools as well as identification tools, quantitative analysis tools, and 21 CFR 11 compliance. And the Duo, I want to show it to you. I want to show you what data looks like, right? We can monitor, that fingerprint and say the stretching region, right, or moisture in the same time phase. So here I've got an example, right, of creating beef jerky and monitoring that with an iRaman Duo system, and we're able to assess, right, both chemical and structural changes, say during this drying process, right, where we can actually look at the aromatics and the amide bands, but also come back and do some deconvolution to understand what's happening with the aromatics in the stretching regions. If we look at, say, our aromatic CHs, our methyl groups, and even the NH stretches that are present inside the system, so we can build a more complete picture to understand what's occurring. And we can apply that, right? Here's another example, right? We want to maybe look at an entire plant. But I'm just giving you the ideas of the things that you can see if we are looking inside. With a duo, we can see really everything in one spectrum. So to summarize, I think I've given you just some examples of how Raman spectroscopy identifies materials and can help you understand changes that are going on. It's got, of course, as we all know, really great promise as both a development tool for developing assays and analyses, as well as actually diagnostics. And we have a new product, the iRaman NXG, and especially the Duo, that extend traditional Raman capabilities using using sequential dual laser excitation. So you get more complete chemical insights by collecting both fingerprint and stretch region. You can then use that to improve the accuracy or reliability of your interpretations, because you've got cross-validation now between regions to reduce ambiguity. Of course, the Duo is a longer wavelength Raman system, so you've got higher fluorescence tolerance that lets you overcome some of the backgrounds that are present in our complex and biological samples. And of course, it's Raman spectroscopy, so it is real-time and non-destructive monitoring. And with that, I thank you for your time.
Thank you, Adam, for that excellent talk, and thank you to all of our other speakers that have participated in today's event. I want to thank the audience for attending and for participating in the afternoon session of this virtual symposium, Emerging Trends in Pharmaceutical and Biopharmaceutical Analysis, presented by Spectroscopy, the Society for Applied Spectroscopy, and the Koblenz Society. Thank you again to our sponsors, CEM, PerkinElmer, Thermo Fisher, Metrome, and Endresen Hauser. We want to remind you to take advantage of the resources from those sponsors, which you can find at the bottom of your screen. If you would like a certificate of attendance for this event, please send an email within a week of the event to training@sas.org. That is training@sas.org. You will receive an email alerting you when this virtual symposium will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. And we hope to see you all next time. Thank you very much, and goodbye.